Going to open for just a few points of discussion, some questions, a short panel before we break. If we can get you guys back, we obviously have an exciting number of talks that were presented today, all the way from Dr. Weiss on the new dimensions of patterning, all the way from Dr. Steinberg on human neural stem cells, John Yu and oxidative stress-induced nano-pro drug, and how after that we had Dr. Kroll and how gold nanoparticles enter the brain, and then ended up with Dr. Janowski and this wonderful talk on the work that is also being done part between an international collaboration. So we can actually open for some questions, please.
Thank you, Alfredo. You left out the good talk that you did.
Is Dr. Steinberg in the audience?
He had to leave.
Yes, okay.
He's back operating already, probably putting some nanoparticles in there.
Alfredo, this is a question for your talk, actually. Very lovely presentation with the migration difference between the normal brain and within tumor. I just wondered whether you can actually expand on how you're dissecting out. It appears that it was more aggressive, the migration, whilst it was within the background of the tumor tissue. How much of that was just the integrity of the tumor as opposed to normal cells is less robust, and also the sort of intrinsic factors, the environmental factors, that's different.
You're talking about the two movies that I showed, where I show cells that are from these are their own cells. So these are cells, human cells implanted back in their own brain, but we're keeping the brain alive in the laboratory. We can keep it alive. And it was a striking difference, and we're quantifying that right now, actually. But it's clear as water that when you put human cancer cells back on tissue that has the microenvironment to promote aggressive behavior, they migrate like crazy, and you saw those movies. It is a combination probably of the microenvironment, and we're trying to understand that, and also obviously probably the cells, how they interact with the tissue. But I think that somehow they get cues not only from the vasculature but from the other cells, and they're able to go through the extracellular matrix in a much more aggressive way. We're in the process right now of understanding some of the chemokines that are potentially responsible for that. But that all right now it took us about 5 years just to establish that model of human cells migrating in human tissue. It started with one of my students, Kaison, who's now one of our residents, 5 years ago, and getting tissue from the operating room into the laboratory. And we started keeping it alive for 1 week. Now we can go for months keeping it alive, but it's taking a fair amount of work. So very, very early stages. But I think that there's going to be dynamite in the future in understanding migration of human cancer on human tissue. Please.
Any questions?
Alessandro, I see you have a question. That's okay. No?
No, well, yeah, a question for John about the activations. That's a very interesting concept and everything. Tell me a little bit, I don't know if it was clear the repercussions of that human applicability about the toxicity of these, radicals that for these concepts are very, very exciting. If you can expand just a little bit, John.
Sure. We know that there are specific environments where there's a high amount of reactive oxygen species, and these are in tumors as well as areas of neurodegeneration. And so we see it as a platform to deliver agents to areas of injury like tumors. And so I think the problem with chemotherapeutics is that it's just washed out in the brain or anywhere else in the body. So we're relying on several aspects of nanometer-sized molecules to the EPR effect to localize in tumor, which we clearly demonstrated with our fluorescent nanoparticles. The larger surface area that can interact with these reactive oxygen species. And the prodrug effect of these being released in cells that have a high amount of reactive oxygen species. So when you look at the concentrations in the tumor versus in kidney or liver or other organs where normally one would see chemotherapeutics, it seems to be highly found in tumor tissue as compared to liver or kidney. So Presumably, it'll be safer, but, those will require extensive animal studies and toxicity
John,
studies.
and just a follow-up question, what about the olfactory bulb? Did I see that correctly in a normal brain, that there was accumulation around the olfactory bulb and tract, or was I mistaken in that?
I don't think I saw that. It was
Okay.
pretty much all localized to tumor, and then there was some in the superior sagittal sinus.
All right.
Yeah.
Another follow-up along that line, there's some data from Earth's lab suggesting stem cells have lower ROS. There was a Nature article about that. Is that generally true for cancer stem cells versus the more differentiated? If that's so, then how would you overcome that in this kind of a strategy?
That's a good point. I'm not sure how that would affect the efficacy of the drug in cancer stem cells. Hopefully, the fact that it would be highly concentrated and not be kicked out by multiple drug resistance genes will help. And so hopefully one could overcome it by a concentration effect. But I guess we'll have to see.
I'd like to add something to that, because if you look at that paper that Jordan and our group produced, where we looked, it's a different compound. So there are people, obviously, like Jordan, who trained with Bob Langer. They're super experts in all types of compounds, and it seems that depending, obviously, on the type of compound that you use, they have certain affinity for certain type of cells. I don't understand. Probably Dr. Weiss can actually tell us a little bit more about that. But it seems to me that they have certain affinity. If you look at those compounds, Jordan looked at thousands of compounds. It seems that some of them have a better affinity to brain tumor cells that grow as spheres as compared to those cells from fetal human tissue that is grown as spheres.
Right. I mean, localization is the issue, but the second issue is this is not released unless there's ROS. And so if there's no ROS, so the question is what's the therapeutic window? So
That'll be a good thing to test in vitro.
So, great session, great talks. My question is for Dr. Weiss. You mentioned something about the conductivity of the molecules. So maybe you already mentioned this, but so is it important to measure these, and are you measuring the conductivity? And can you use, say, the change of conductivity to your benefit?
So there are really two parts to that. In the switches and some of the other systems that we look at there, the conductivity is central to the issue, but not, as I said, not really to replace silicon, maybe as some intermediary to go between technological materials and the chemical and biological worlds. In our measurements in the polarizability, one of the things we've been able to do is to map the multiple bonds as a way to differentiate amino acids, and then look at, for instance, beta sheets and helices and where they come into contact with a substrate and each other. You get some enhanced polarizability because of that contact. When you look at the switches as an example, you can see that so polarizability is just response of the electrons to oscillating field, and you can see that the contribution from the metal underneath is as big as the contribution from the molecule's electrons in the on state, and it changes the factor of 2 in the off state for exactly the same molecule. So that tells you, number one, you're not measuring a single molecule, you're measuring the whole system, but it also tells you that contact is for the purposes of determining structures, useful contrast agent within the molecule, if you want to call it that. And so part of the reason we showed those sort of coloring thresholds was to try to differentiate what intermolecular contacts look like as we start to explore this microscopy versus the molecule substrate contact.
Okay. So because I was just wondering if there'd be any use, like say, as you're doing this STM imaging, you could maybe try to inject some electrons, and that get a good measure of the conductivity and change of conductivity.
So, yeah, well, the electrons are essentially transient in that they're going through the molecule, but it's true if you change the oxidation state, you can observe that, if you have a way to hold that charge in the system.
Yeah.
And thanks for your help with the display properties, too.
No problem.
Very much appreciated, yeah. Okay, I'm very good.
Dr. Janowski, was wondering what is the minimum number of cells that you could detect, And then if those stay in a bolus, you can detect them, but over time, what if they're going to migrate out through the brain or migrate through spinal cord? How could you differentiate loss of signal as either being cell death or in fact just that the cells are leaving that one nidus? And the last question, follow-up, would be, what magnet were you using, and would a greater strength magnet enhance your ability to detect the signal?
Okay, so for preclinical studies, that was 11.4 Tesla. But in general, you can see a small number as like 100 cells. But the problem is that you can, with MRI, you can only see the delivery engraftment over like maybe 1 day that is useful. Then you can only see that the label that you labeled with the cells is still in this area, but cells can be died. So that is, you need multimodality. I mean, now you need because it must be something like bioluminescence when substrate due to ATP is transferred to the luminescence product. If there is no, because now all the systems like ferritin in labeling doesn't work for this.
And I'd like to follow up with a question for that, Dr. Janowski. You mentioned that that patient specifically, and this is very important, improved. There was some improvement, but you can't tell whether it was due to the fact that nature, the patient is getting better, or your treatment. Can you comment in respect to that, please?
Yeah, that is very difficult. So what we could do, we put them first for 3 months for very regular rehabilitation, and we followed them over 3 months, and we didn't see any improvement. So that's why we decided at all to transplant cells. That was initiative of the family of this kid, because that was pretty old parents, and that was very difficult to have this kid. So that's why that was very, very important for these parents. And they found us. They wanted to transplant the cells, but so we have a 3 months observation, no improvement. Then transplantation. And some improvement, what we found was over first 6 months, and then what we found after 6 months, there was no further improvement over next 2 years now, because there is 2 years observation, and so it can be due to the cells, maybe due to the trophic factors. There was some study from Italy published that they injected only NGF. With a pump to the ventricular system, with the very similar situation, also 1-year-old kid, and they have only some improvement. So it can be due to these cells, and we use all of the cells that was frozen from this kid. So we don't have more cells to transplant more.
Thank you.
Just wanted to make a final comment on the session. The society will have a press release about the importance of integration of nanotechnology with stem cell and medical devices and medical imaging, and that's the theme of the annual meeting, and that's the area that Brain Mapping Foundation is going to push for National Center for Nanobioelectronics. So in that regard, I want to ask an opinion of the panel on that, and see if we can reach a consensus that this is the way to go. From Dr. Weiss, you're first.
Well, certainly we hope control of chemical functionality and will, number one, enable us to understand and probe, and then ultimately control interactions, right? So if we can direct stem cells, if we can use those interactions to advantage, I mean, certainly where we'd like to be able to go with what we've been working on.
So you agree that integration is the way to go?
Yeah, we have a long way to go, but we have the means to control from our side, and now the question is, can we understand the biological interactions sufficiently to gain that next handle? Yeah.
Well, I think the whole field of stem cells has really given us great insights into the initiation of cancer, propagation of cancer, of neurodegenerative diseases, and so it's given us great insights to those. It's given us some flavor of potential therapies, as has been discussed, and nanotechnology clearly is a nascent field with great potential as well. So I think it makes sense to put them together. I think it's going to be a huge combination of potential, interactions, but I think it deserves, some impetus to develop.
In my opinion, I think it's very, very important. What was the only thing I was missing a little bit, that nanotechnology is somehow still, not so many nanotechnologists were here on the conference. So I think for the moment they don't know, but I think it's extremely important to include them, because I think there will be in the future, especially for stem cell delivery, marking, imaging, they need to know what is the needs in order to tailor this. And so I think it's perfect to include them, but I think it's not yet well known that this conference exists.
I think that all of us are working really in the interdisciplinary area, and such a collection of people gives a chance to have some new inspiration of new direction of research. So all of us show what now it's feasible, what we can do now in our labs. So that it's very important, and it's after sometimes after 1, 2, even week and months, then we took something from these presentations to our project, for example, and then that will be much better. So that is, in my opinion, that is very good thing for the people that are really interested in the interdisciplinary research. What we do, in molecular biology, we engineer cells, and then we see what's going on in MRI. We see what's going on with the behavior. That's all, and also we're going with clinic to this. So that's beautiful session for me.
Thank you. And Dr. Quinones, your final thoughts on that?
Thank you, Babak. I am reminded of the Nobel laureate Santiago Ramón y Cajal, who won the Nobel Prize in 1906 with Camilo Golgi. We saw Golgi stain right here, and he said once that before we can understand and go out there and understand the universe, we ought to try to understand the universe that we can hold in our hands, and that is our brain. And I really do believe that through our utilization of the future technology of stem cells at a cellular level, as well as nanotechnology, that is within our reach. And I think within our lifetime, and I'm talking the next 2-3 decades, we are going to have major advances in this field. And I think this is the time to commit our resources to do so. Thank you.
Thank you. The meeting is adjourned, so we'll go for a break. Thank you.
Thank you.